Boyle's law, in the case of where we get negative pressure in comparison to the intrapulmonary, which is on the inside of the lung, compared to the outside air. That's how we're able to get air in. If we have positive pressure, meaning higher pressure in the lung compared to the outside, that'll get air out. So what we want to be able to do, especially in the clinical setting, is to measure the lung volumes, how much air is moving in and out. And we can do that by measuring static volumes of air. And some of the old ways of looking at it, you can see if you take a look at around the room, you see those blue tubes or those blue canisters in the back. And tied to those blue canisters is a string that's attached to basically measuring the amount of air, or technically water, that you can displace. And these are the old school spirometers that are used, lung volume spirometers, where you get somebody that closes up their nose. They're going to exhale into this machine, and this will cause the pressure to increase, and the pressure increase will cause this piston to rise. And the amount of rising that's causing the piston will get recorded on a device that will basically be able to measure how much volumes of air are we able to inhale as well as exhale. In these spirometers, they would typically be used to exhale the amount of air out. So this helps us measure how much air is breathed out during different phases of breathing. What we're going to be using in the lab on Thursday, we're going to use these small spiropets. And basically what it is, instead of using those old tubes right there, because I've had people try to exhale, I've had people try to inhale, I've had people that will cause water to splash around. We don't use those things anymore. We'll use a device that will basically be this device right here with a little turbine on the inside, and the number of times that turbine spins will be able to tell us how much air is being breathed out. Not as accurate as these things, which will actually capture the amount of air exhaled, but nonetheless still give us somewhat accurate measurements. So how do we measure our spirometry volumes? It's typically using these our spirometry volumes. And this plots out, the amount of air, of which you can see to our left will show you the milliliters, the number of milliliters of air, and from the top of our breath all the way down to the bottom of our breath. So these lung volumes can be easily explained. The first point which I'm going to point out is quiet breathing, which takes place around, and the volumes that you're seeing here are, again, just generic volumes that I'm using. These are actually going to be adjusted based on your height, your age, your height, your age, as well as your gender, and then your ethnicity is going to play a factor on how big your lungs are. And what we described as quiet breathing is this segment right here. Here you can see any movements upward is going to be inhalation, and any downward movement is going to be exhalation. So during quiet breathing, which you are all doing right now, all of that is being autonomically regulated, which we'll talk about autonomic regulation at the very end of this entire respiratory lecture. Tidal breathing is going to show you quiet inhalation, quiet exhalation, inhalation, exhalation. And we can measure that volume, which is about 500 milliliters of what's called tidal volume breathing. And why do we call it tidal volume? It's literally like the ebb and flow. It's the tide moving in and out. And that's exactly what we're doing with our lungs. Like I said, 75% of that breathing is due to the diaphragmatic motions, the movement, the contraction, as well as the relaxation of the diaphragm. And that's going to cause that 1 centimeter movement of that diaphragm is causing 500 milliliters of air to go in, and then that relaxation causes 500 mils of air to move out. That's called tidal volume breathing. Now, we're all doing that exercise right now where we're doing tidal volume breathing, but I want you guys to try this out with me. So I'm going to guide you guys through normal breathing, which you guys are already doing now, and then I'll tell you at the top of your normal breath. At the top of your normal breath, what I want you guys to do is to breathe all the way to the very top. So whenever you're ready, the top of your normal breath, take a deep breath all the way to the very top. Okay? And go ahead, start breathing normally. Once you get to the very top, you've filled your lungs all the way. And typically in a clinical setting, you want them to breathe as much as they can until they fully expand their rib cage. Right at the very top is what we call total lung capacity. That's how much air your lungs can hold. and you filled it up to your maximum. From the top of your normal breath, which would be, and let me just change the color here because I don't like the color scheme. At the top of your normal breath, that was your normal quiet inhalation. I had you guys breathe all the way up to the very top. That section from here to here is your inspiratory reserve volume. So think of that as if this was your tidal inhalation, and then I said, okay, now that you've reached the very top of your normal inhalation, keep breathing all the way to the very top. This is what's tapping into your reserves. This is your ability to breathe all the way up to total lung capacity, also known as TLC. And when you see all these explanations that I'm doing, I've got that listed all out for you right here on the next slide over. So inspiratory reserve volume. Now, the ability for you to breathe from the bottom of your normal breath. I just exhaled, and now at the bottom of a normal breath. So inspiratory reserve volume plus your inspiratory tidal volume breathing, that one inhalation here, that's your normal breath in, and then all the way up to your inspiratory reserve volume, all the way up to TLC, is what's called our inspiratory capacity. It's your capacity to breathe in from the bottom of a normal breath. So one of the things of what we could do to figure out what inspiratory capacity is to take tidal volume plus your inspiratory reserve volume. That's everything from up here down up to the total lung capacity. Now at the bottom of your normal breathing, let's have you guys go again back into normal breathing, which I know you guys are already doing. At the very bottom of your normal breath, I want you to breathe and squeeze all the way out and squeeze your lungs completely. And once you get down to that very bottom, go back to normal breathing. So what have we done? We've tapped into what's called our expiratory reserve volume. It's from the bottom of a normal breath, and you squeeze all the way out, and you're going to squeeze out as much as you possibly can. And we've tapped into, like I said, our expiratory reserve volume. What's left over is not 0 milliliters of air. There's still some leftover air that's still residing in your lung. And what is that called? That's called the residual volume, RV. And that's what prevents, the leftover air is what prevents the airways from fully collapsing. You've squeezed everything out, you've compressed as much as you can. This is going to be reflective, and which we'll talk a little bit later. Residual volume is going to be reflective of how much air is left over in the conducting zone. Because we can't really squeeze everything out. I can empty out my alveoli, but I can't squeeze the trachea, I can't squeeze our bronchioles. Some of that air is left over, and this is again called our residual volume. Now, from the very bottom of your breath, all the way up to the very top of your breath, when you're at TLC, is what is called our vital capacity, VC. And VC, which we define on the next slide over, all the air that can be exhaled after a maximum inspiration. So the way of measuring this is to get somebody to breathe all the way in, all the way to the very top, and exhale as much as they possibly can. And that vital capacity is usable airspace. It's the capacity that's vital to life. And so if we were to take vital capacity and add the functional, or add the residual volume, that's going to equate to our total lung capacity. So with this piece right here, residual volume, there's no real way of being able to measure that with any of these machines. There is a machine that you can actually use called a body plethysmography. It's basically a chamber. It's a telephone booth looking thing that you go into, and it locks you into this chamber, and you're inside this little chamber, and they have you do some normal breaths. And once they get you down to the bottom of a normal breath, they lock the chambers, they lock all the pressures, and they have you kind of move, kind of mimic some diaphragmatic breathing, and that's going to cause minute changes in pressure in the lung, which will also cause some minute changes in the chamber, and from that, due to Boyle's law, we can infer how much air is left over at the bottom of a normal breath, which will give us the number for our functional residual capacity. And the functional residual capacity basically comes down to, and this is at the bottom of your normal breath. Your FRC is a combination of your expiratory reserve volume as well as your residual volume. It's the volume of air that's left over at the bottom of normal breathing. Okay? That's normal passive exhalation. Okay? And how we passively exhale from that point, we utilize, again, the same principles. Surface tension of the alveolar wall. We use the connective tissue surrounding the pulmonary lobule and the elastic recoil of the chest wall. Those are the principles that guide functional residual capacity. So all of that, again, gives us the amount of air that can move in and out during these different phases of breathing. Any questions on this? Yes, Ellen.
Inspiratory capacity was the tidal volume plus the what?
Inspiratory reserve volume, IRV plus the tidal volume.
IRV plus the tidal volume.
Yeah, so tidal volume is normal breathing, and the inspiratory reserve volume is what you breathe in after you've taken a normal breath. Okay, so that is your capacity to inspire. What other questions?
Will we have to remember all of these for the test?
Well, if I were to pull up a chart, if I were to pull up a table or have all this out there for you, you should be able to identify which ones are which. So here are your equations. Again, next time we take the test, which is next week.
Tuesday.
Wait, today's only Tuesday? Damn, okay. My days are always off. Next Tuesday, remember to bring in a regular calculator. So don't forget, you're not borrowing a calculator, so make sure you run in, grab a cheap calculator, no cell phones, but something that can easily do some multiplication and addition. Nothing complicated, no TI-83s, regular calculator.
Can you send us a reminder email like the day before?
As long as you can send me a reminder email to send you a reminder email. But I'll making a mental note now. All right. So with that is our volume. So this is the ability to give us normal volumes. And like I mentioned earlier, the easiest way to remember this is think about your normal breathing and then think about your reserves. And from that, you'll be able to kind of deduce all the other ones. Vital capacity, total lung capacity should be fairly easy. Inspiratory capacity, look at the name and they should be easy to explain. The other one I just wanted to explain for you is functional residual capacity, which can throw off some people sometimes.
Do we need to remember all the number?
The numbers, no, not at all. The numbers are not important because these volumes are based off somebody's actual calculation. These are just generic numbers that I'm throwing out. Okay.
Thank you.
So typically these would be things off, they use just a standard measure, some 75 kilogram male, and these would typically be the average measurements from them. But there's a lot of variation on the size of somebody, the genetic makeup, height, etc. So with that, we've talked about static volumes. Again, that's the volume of air that we were able to move in and out. And again, we'll be doing that on Thursday during our labs. Now, what we're going to be looking at is ventilation measurement. While earlier we were able to measure the amount of air, this doesn't necessarily tell us the speed, or not really the speed, but the volume of air in a given amount of time. So we want to start looking at this dynamically. How much volume of air can we get in when we account for time? Okay. So when I say do normal tidal volume breathing, I didn't say breathe at 20 breaths per minute or 15 breaths per minute. What we want to take a look at in ventilation is accounting for that static volume over time. So that equates to the rate of air movement. In other words, how fast am I moving that accordion to get air in and out? And the way we measure ventilation is the liters of air per minute. Okay? So how we measure ventilation is to look at the volume of air, right? The volume in terms of liters per breath, times the respiratory rate, how many times you breathe per minute. So if I were to take a look at my last slide shows me tidal volume. If I'm going to be measuring your ventilation, the amount of air that you're breathing per minute, also known as minute ventilation, I want to know how much are you breathing during quiet breathing times that by the number of breaths that take place in a minute. So how does this work out? Liters per breath times breaths per minute. Cancel out the breaths, and you get the amount of air in liters per minute. This equation should look like something that you saw earlier. Very similar to cardiac output, where cardiac output was the amount of blood per beat, and then how many beats per minute, where volume would be similar to that of your stroke volume, and the respiratory rate would be similar to that of your heart rate. So in essence, minute ventilation, which you see down below, minute ventilation is your tidal volume times your respiratory rate. And again, that comes out to how much air is moving in through the lungs per minute. That's part of the equation what we're looking at. Again, ventilation is just the movement of air. And so, as I mentioned earlier, when you were able to get down to residual volume, there was some air that's left over in the lung. That comes out to something which we call anatomic dead space. And anatomic dead space is the amount of air that's left over in the conducting zone after a full exhalation. Okay? You've emptied out your lungs completely, and you've still got some air left over in the trachea, some left over in the larynx, some stuck in the pharynx, some still stuck in the nasal cavity, down to the trachea, down to the primary, secondary, and tertiary bronchioles. That is the total volume of the conducting zone. And how much does that come out to? That's basically about 150 milliliters of air left over. And how do they know that? Again, they've done it on cadavers where they'll take the conducting zone and they'll fill it up with water, measure the amount of water. It comes out to about 150. And again, that varies based on your height, your gender, as well as your ethnicity. So looking at that, what this comes out to, as I mentioned earlier, the amount of air that's left over in the conducting zone is about 30% of normal quiet breathing. The other 70% makes its way to the respiratory zone. So during normal quiet breathing, about 30% of about 500 milliliters of tidal volume breathing. That comes out to roughly 150 to 175 milliliters of air that's left over in the conducting zone during normal tidal volume breathing. So the question is, why that waste of air? Why is it just sitting in there, and what's the whole purpose of it? Well, some of that is going to be used to humidify the air, again, filter the air, and moisten the air. So there is a purpose for that. So think about what's going to happen as you start to help visualize the inflation of the lungs, when we're inflating the lungs to fill it up with air, and the movement of air molecules that are following through. So I'm just going to quickly just draw, just for the purposes of demonstration. Anytime you take a breath of air in, you can see this whole static column of air molecules moving in. And what happens when we take a normal breath in, let's say we take normal fresh air in, air is going to move into the lung. 70% of that is going to make its way into the respiratory zone. The other 30% are going to get stuck in the trachea. So when somebody takes fresh air in, we would expect all this air to be the same composition of that of outside air. It's fresh air that gets trapped in the airways. And when somebody exhales the air out, they're going to be able to push some of that air being fresh air, and once you completely empty the lungs out, now you've got all that fresh air out, you're going to continually push old air out. And at the very end of your exhalation, about 150 to 175 mils of old stale air is going to sit in your lungs, or sit in your conducting zone. So think about it, at the very bottom of your breath, the air that's left over that you're finally exhaling out is the stuff that's left over, the residual air that, well, I don't say residual air, but most of the air that you've been able to evacuate out of your lungs. And then when you switch and take a breath back in, you're rebreathing some of that old air back in. And I'll talk about that in the next slide over. So before we get into that, let me just kind of continue on with that concept, with the whole string of air molecules that I'm showing. On the next slide over, we've talked about ventilation, of which 70% makes its way into the respiratory zone to participate in external respiration. The other 30% remains in the conducting zone within the airways and does not actually participate in gas exchange. So with that, we want to be able to measure ventilation and perfusion. So ventilation, we know about 70% makes its way to the respiratory zone. 100% should be perfusion. Again, blood flow. This represents, ventilation represents the movement of air. Perfusion represents the movement of blood. And as I mentioned in the beginning of class, we want to make sure that the amount of blood flow through is optimal for that of well-ventilated alveoli. So we want about 70% of the air to make its way in here and 100% of good blood flow around the alveoli. So the normal ratio for ventilation to perfusion ratio is 0.7. That's 70% divided out by 100%. So that normal value is about 0.7. Why is this important? It's some of the things that are also where we're able to discern any abnormalities of the lung itself. So what we consider VQ mismatch, these are numbers moving away from 0.7, of which, let's take a look. As I mentioned earlier, you're going to have normal ventilation, which is going to be 0.7. We're going to have normal perfusion, which is 100%. What happens if ventilation is completely blocked? Okay, so let me take a blockage in this alveoli. I've got no good air, not no good air, but not fresh air.
No air.
No way.
Old air, there you go. We'll call it old air that's trapped in the alveoli and does not exchange with the outside environment. So I've got old stale air in there that's low in oxygen, right? And then I have normal perfusion. I've got normal blood flow that's flowing through. That should be obvious there that this is a waste of perfusion. This is blood that's getting directed to areas that have poor ventilation. So therefore, that's going to reduce, if our VQ, and it's actually not P, it's Q, I apologize. VQ, where our normal ventilation is decreased and our perfusion is normally 100%. So therefore, our movement, our VQ ratio at this point where we have zero ventilation comes out to 0 divided by 100, comes out to a ratio of 0. So when we're moving a VQ ratio towards 0, that's letting us know ventilation is down, but perfusion is normal. So somebody who has this, why would this be important? Well, think about what happens with pneumonia. Pneumonia is going to be a fill-up of fluid, so the alveolar space is going to fill up, and now you're going to block the alveolar interface from that of normal capillary function, meaning blood flow is normal, but no ventilation. The converse of this is, actually, I apologize, we're going to, what we name this is called shunt. And the shunting of this is where the alveoli are filled, or we're not allowing normal ventilation. Typically what we would need to happen at this point is to redirect that blood flow to areas that are well ventilated. And I've got a couple slides that will talk about that. Now, the opposite of this is where I get normal ventilation. Take this alveoli, and I can get normal air movement. I got normal ventilation in the alveoli, but I've got no perfusion, meaning blood flow is decreased around the alveoli. So in this case, I have I have ventilation at about 70%, which is normal, and perfusion would be 0%. So anything, a number divided by 0 is undefined, about infinity. So in this case, this is what we call dead space. Dead space, in a sense, means I've got air that's able to go in, but no gas exchange, no external respiration taking place. So in other words, I've got pockets of air in the lung that are not participating in gas exchange. So this is considered you don't need to know the term of it, but it's considered physiological dead space. Anatomic dead space is what's trapped in the anatomical structures of the conducting zone. Physiological dead space is due to poor perfusion. And looking at these, typically what would decrease the amount of perfusion? Pulmonary edema. Okay, so pulmonary hypertension can create an excess of fluid that builds up. Now, to tie it back to our earlier lectures in the very beginning of our course, I talked about things that would decrease the diffusion of gases across spaces. So here we see gas exchange taking place. O2 is supposed to cross into the capillary, and CO2 is supposed to move out from the capillary to the alveoli. If I can decrease diffusion by reducing the amount of surface area, which isn't really physiologically relevant, but if I can increase the diffusion distance, that's going to effectively decrease the amount of perfusion, the amount of blood flow into this area. I can also take a look at pulmonary hypertension, which will affect the amount of perfusion, things that interrupt the blood flow. Any way you look at it, it's a disruption of both either the ventilation or the perfusion that will result in various lung problems. The inability for external respiration to optimally take place. And that physiological dead space I described for you over here. So this slide I probably need to move over back one. But this is what we call physiological dead space. This is properly ventilated areas of the lung that does not have perfusion and will not participate in gas exchange. This is what we want. This is what we call VA. It's alveolar ventilation, where that measures the amount of actual fresh air making its way in and good perfusion to that area. So the other concept of anatomic dead space, I'm going to go back to this example that I was drawing out earlier, is the column of air that's moving into the lungs. Our anatomic dead space, what actually happens during normal breathing? Again, we're going to assume tidal volume, VT, to be 500 milliliters of air. That's normal breathing, 500 milliliters of air moving in or out. And at where we're going to start off with is your lungs filled. You've filled up with normal air, and you've got 150 milliliters of fresh air that's sitting in the conducting zone. As I mentioned earlier, you've taken a 500 mil breath of air in. 150 is going to be sitting in the anatomic dead zone. Now, what happens when we exhale? We're going to push out 500 milliliters of air. That means 150 milliliters of air that was trapped in the dead space is going to get pushed out into the airway, into the external environment, and the other 500 is what we exhale. 350 that's going to get expired out into the air. The other 150, which is going to get trapped in the dead space. As I mentioned earlier, when you squeeze out at a normal exhalation, some of that stale air is left over in the lungs. Now that you've squeezed that out, 150 is staying in the lungs. What's going to happen when we take another 500 mils of air? This 150 is going to get pushed back into the alveoli. While the other 500 that we take in, 350, again, 70% making its way into the respiratory zone. The other 30% being trapped in the airways. So any way you look at it, there's 150 milliliters of air that's going to get some old air that's going to get continually pushed back into the lung. This is where you're going to see mixing of fresh air with stale air. And there's a purpose behind this, because that stale air we'll be able to see is all for the entire purpose of keeping, obviously keeping the airways open, but that mixing is going to be physiologically relevant. So we'll be able to see what's going to happen in terms of the partial pressures, the concentration of the O2 and CO2 as it mixes with that old air. And I'll bring that up in the tail end of today. What questions do we have about this concept?
So basically it's a circle, right?
It just goes back
It goes
around.
back and forth.
So all it comes down to, is you're breathing 150 mils of stale air every time, of that 500 mils of fresh air. So one of our last equations, I believe. I think. All right, so what we've mentioned earlier in our prior slide is how we measure ventilation. And in this ventilation, we've been able to look at tidal volume, which we count as 500 mils of air, times the respiratory rate. So in this equation, let me take a look, and we did 6,000 milliliters, which comes out to 6 liters of air. 6 liters of air equals 500 milliliters of air. Come on, now, let's do your math here. 500 mils of air times 12. Yes, thank you. That was right there. 12 breaths per minute. So that, looking at this, gives us our 500 mils of air. 500 mils of air times 12 breaths per minute comes out to about 6,000 milliliters per minute. So that is something that we want to look at as normal ventilation. How much air are you breathing in and out of your lungs in a minute? Comes out to about 6 liters per minute, again, generically speaking. But what we do need to account for is this: 70% of your tidal volume reaches the respiratory zone. So what I need to account for is how much of that air, of that minute ventilation, is making its way into the alveoli. So naturally, what I'm going to have to do is take that tidal volume, which we know to be 500 mils of air, and times that by 70%. So let's take a look at the equation for alveolar ventilation. And alveolar ventilation does exactly that.
The last equation, what was it for? The last one?
That was minute ventilation. So minute ventilation is tidal volume times the respiratory rate. Now the alveolar ventilation is taking this component and accounting for the dead space. And the dead space we already determined to be about 150 mils of air that we saw in that prior slide. 150 mils that are stuck in that dead space. So with our alveolar ventilation, we're able to account for the dead space, and this will tell us how much air is making its way to the alveoli, which is going to be representing the fresh air that's going to participate in external respiration. So what I want to do, as I mentioned earlier, is to take a look at our quiet breathing at rest, given the example I mentioned earlier. I have, you have a question? Tidal volume of 500 milliliters of air per breath, times the respiratory rate of 12 breaths per minute, and accounting for the dead space volume, all I'm doing is doing 500 minus the 150 comes out to 350 times the respiratory rate of 12. And if I do that, 350 times 12 comes out to 4,200 mils per minute that's making its way to the alveoli. Again, accounting for that dead space. If I were counting minute ventilation would be 500 times 12, and that comes out to 6,000. So if you do 4,200 divided by 6,000, that should come out to 70%, right? So there we go, 70% making its way to alveolar ventilation. Now let's take a look at the other breathing patterns that could play a role on alveolar ventilation. And let's take somebody who does deep, slow breathing. So what would we expect with deep, slow breathing? We should see an increase in their tidal volume and a decrease in their respiratory rate. So we're going to adjust their respiratory rate to 5 breaths per minute and their tidal volume to 1,200. So now I'm going to do 1200 minus 150, which is going to be 1050, times that by 5. And that comes out to 5,250 mils of air per minute that's making its way to the alveoli. Now let's do the other way. It's going to take somebody who's going to take some shallow breaths, but really rapid breathing. So we're going to expect a decrease in the tidal volume and an increase in the respiratory rate. So that's somebody that's breathing like this. Now take that at a tidal volume of 150 mils of air. 150 minus the dead space of 150 times 40 breaths per minute. And that comes out to 0 mils of air, alveolar ventilation. So this should explain what we need to do when they tell you, take a deep breath in, slow your breath down. That's going to increase your alveolar ventilation, which is going to essentially bring in more fresh air into the alveoli. So it's more effective to take deep, slow breathing. In other words, increase your tidal volume and reduce your respiratory rate than it is to hyperventilate with small tidal volumes. Because imagine what you're doing in that case of 150. All you're doing is taking that 150, it's going back and forth between the anatomic dead zone, and eventually you're just going to run out of O2 that was originally in that sample of air in your lungs.
Is that the reason why you write shallow breathing and right it?
Exactly, because what's happening is it's hypoxic. Technically what's happening is you're also hypercapnic, meaning you have high CO2 and low O2. And eventually, when you get low O2, your partial pressure of O2 drops, and you're not getting as much air, or you're not getting not as much oxygen into the brain tissue.
And this doesn't apply to dogs, because I know like dogs have like that.
They can, but there's still an autonomic regulation. We still have a way to, like for example, if I told you to constantly do that very shallow breathing, eventually you're gonna your central nervous system is going to take over and say, nope, I'm going to take over, and it's going to make you take a deep breath in. Okay. So you might have heard the whole concept of yawning. I don't want to go too much into that because I haven't really looked at the physiology behind yawning, but it's one way to take a long, deep breath in that's going to exceed that of tidal volume and be able to get fresh air into the alveoli. What's that? All right, so that is alveolar ventilation. And this should explain also what happens during exercise. When we start to increase our pulmonary ventilation, we're going to need to increase our alveolar ventilation. And the best way, again, increase your tidal volume and reduce your respiratory rate, because we want to account for a higher tidal volume than that of our dead space. With that, what questions do we have?
We're going to need to know all of these for the test, right?
If it's going to be an equation.
Good questions.
But really, if you were to look at it, it's not that complicated. Minute ventilation, again, is breaths per minute times the amount of air. All this alveolar ventilation, all it does is it just accounts for that 150 mils of dead space. So subtract that from your tidal volume, times that by your respiratory rate, and you should be able to see. As long as you remember 0.7, I think you're almost there. All right. So without any further questions, let's move into gas exchange. Okay, let's move into gas exchange. And this concept is going to employ all of Dalton's law. And again, Dalton's law, if we go back to earlier slide, Dalton's law is the partial pressure law. Henry's law we'll talk about in just a little bit. So Dalton's law is just basically taking the composition of a particular gas. So if we're looking at oxygen, for instance, we're going to look at the partial pressure of a specific gas in a measure of air. So when we look at an entire vessel of air, whether it's in this room. We know 21% of that is oxygen. We know that the pressure is 760 millimeters of mercury. And for us to be able to measure the partial pressure, like I mentioned earlier, you take 21% times that by 760. And your partial pressure for O2 comes out to 160 millimeters of mercury. And that's our equation down over here. If I were to measure the partial pressure of CO2, the partial pressure is, or the composition is 0.04%. So 0.04% times 760, and that comes out to about 0.3 millimeters of mercury. Very small partial pressure. And if I take into account nitrogen, I take into account water vapor, add those all up, that should equate to 760 millimeters of mercury, or total atmospheric pressure. So what we're going to be doing in looking at Dalton's law is to look at how external respiration is going to take place. How does gas exchange occur? And what we need to do is to take a look at the factors that affect the rate of gas exchange. And these are going to employ the same principles that we already learned, just tied into pressure. Partial pressure difference is looking at the movement of a gas that can diffuse across a permeable membrane from a high to a low partial pressure. Just like you learned in a cell. In a cell, we have high extracellular sodium, and we have low intracellular sodium. We know that there is a concentration diffusion gradient from high down to low, where we get a high concentration down to a low concentration. Gases work the same way. We work from a high pressure down to a low pressure. You've learned about the mechanisms that work for the pressure of air, that move air from outside to inside. We're going to take a look at how partial pressures basically follow the same principle, where we move from a high to a low partial pressure. That's one of the major factors for the rate of gas exchange. The second is to make sure that our ventilation and perfusion are properly coupled, meaning that we have optimal ventilation and we have optimal perfusion. None of those are working, whether it's ventilation or it's not perfusion, then we're not going to get gas exchange to occur. And surface area, the 750 cubic feet that I talked about for the alveoli represents the surface area. So an increase in surface area will increase the amount of gas diffusion that takes place. The diffusion distance. If you remember from our earlier experiment, you guys were the extracellular sodium ions. You guys are the intracellular sodium ions. I've got a cell membrane right in between you guys. And if I thicken that membrane, or I thicken the diffusion distance that you'd have to travel, that would mean there's less of the extracellular sodium ions moving into the intracellular side. It takes much longer, and it slows down the rate at which sodium moves in. Now, applying that, the distance that O2 has to travel from the alveolar capillary interface is going to decrease the amount of gas exchange that's going to occur. So, increase in diffusion distance results in a decrease in gas exchange. And then gas solubility. We have to take a look at Henry's law that accounts for how much of the gases can freely dissolve in a fluid, of which we know that CO2 is much more soluble. It comes out to about 20 times more solubility for CO2 than O2. Hence why we see more carbonated drinks with carbon dioxide than we see oxygenated drinks. So with that, we're going to start to see our partial pressures. And one thing I want to do on here is to note in the alveoli, notice that the partial pressure is 100, while the partial pressure of CO2 is 40. And what I'm going to do is just kind of continue this example and draw it out over here, where this is going to be external air, where we measured our PO2, our partial pressure, to be 160 millimeters of mercury, and our PCO2 to be 0.3 millimeters of mercury. And as I mentioned earlier, due to that dead space, that PO2, which is fresh air, noting to be 160, by the time it makes its way into the lung where it's rebreathed, it's going to be down to about 100, and CO2 levels are going to rise from 0.3 in external down to a PCO2 of 40. So 100 for our fresh oxygenated, and 40 for our CO2 in oxygenated air. Now, what we're looking at here is the direction, in our slide on the image on the bottom right, is looking at the partial pressure movement. Looking at our blood, this would be deoxygenated blood that's moving in this direction. I get deoxygenated blood that's making its way into the alveoli. And we'll note that the PCO2 is 46 and 40 for our oxygenated blood. And for you to get this concept, really it's just the concept of just having to memorize the numbers. 46 for CO2, 40 for O2. Looking at the diffusion, the partial pressure gradient, we notice that PCO2 is higher in deoxygenated blood. So it's 46 in capillary, it's 40 in the alveoli. The direction of CO2 movement would be out of the capillary and into the alveoli. Now, the converse of looking at what happens with O2, it's higher. We've got 100 partial pressure of O2, partial pressure of oxygen at 100 millimeters of mercury, where it's 40 in the deoxygenated blood. So the direction of O2 movement is from high to low, from the alveolar into the capillary. So here, this is demonstrating the exchange that's taking place where we get CO2 pushed into the alveoli, and we pull O2 into the capillary. This is external respiration. Again, because this is communicating with outside air, our external environment. Now, internal respiration is taking that oxygenated blood. And when we see 100 millimeters of mercury and you see 40 millimeters of mercury in the capillary, we're eventually going to oxygenate the blood very quickly up until that the PO2 makes its way and it's 100 millimeters of mercury, okay, and 40 millimeters of mercury. So you're going to see a matching of alveolar partial pressures with that of capillary partial pressures by the time it makes its way through the systemic circuit before it arrives to the target tissue. So now we see 100 and we see 40. We take a look at an active cell. Let's say this is a muscle cell, and this muscle cell is using that oxygen. So it's using oxygen. It's going to have a low partial pressure of O2. So 100 over to 5. The direction of movement of oxygen is to move from the capillary into the cell. And then the direction of CO2, the CO2 that's being produced during glycolysis, during the Krebs cycle, all that CO2 is going to diffuse out from the mitochondria into the cytoplasm. CO2 is going to now diffuse out into interstitial fluid into the capillary. So this gas exchange where O2 is moving in and CO2 moving out on the tissue layer is called internal respiration. And here you can see, now we're able to extract some oxygen from that, and we're going to turn it back through, switching back to external respiration once we make our way back to the lung. What questions do we have about that concept? We'll be able to see that in this slide over. Not much more complex, it's just drawing it out a little bit more widely. So now that we've talked about the partial pressure gradient, let's take a look at the mechanics in external respiration. And looking at the partial pressures that take place within the alveoli and the capillary. So as I mentioned, that partial pressure gradient for O2 is moving from 100 down to 40 of that in deoxygenated blood.
Why does the pressure decrease? Is it because of distance?
No, that's just the oxygenated blood. Take a look at the, let me jump to a later slide. This slide right here shows you the partial pressure of O2 in oxygenated blood. by the time it makes its way and it passes through the tissue, by the time it pulls out, it's able to pull that oxygen into the cell, which is going to reduce the amount of oxygen in the capillary. So that by the time we make it back to deoxygenated blood, our PO2 is about 40 millimeters of mercury. So in essence, to answer your question, we're pulling, in deoxygenated blood, we're pulling oxygen away, and we're going to replenish that in external respiration, meaning we're going to replenish that back in the alveoli. So while we have 100 millimeters of mercury of oxygen in the alveoli, we've got a partial pressure of 40 in the capillary, oxygen is going to diffuse across. And that O2, here you can see that on our slide below, that partial pressure is about 40 of deoxygenated blood. Here you can see in blue. And we're going to equilibrate to bring that blood all the way back up to a PO2 of 100. And how long does that take? From this point to this point is going to be the travel time for a red blood cell to make its way from the venous side, I'm sorry, the arterial side of the capillary bed to the venous side of the capillary bed. Here you're going to see the start, again, the arterial side to the venous side. And the time that it takes, and again, you don't need to know this, the time it takes is about 3/4 of a second to make its way from here all the way to the very end. That means that the equilibration takes place within the first third of its transit time through the capillary bed. You watch from 40, and we equilibrate back up to 40. I'm sorry, back up to 100. And that's that second point right there. Now, carbon dioxide moves in the exact opposite direction, where we're pulling CO2 from the capillary of deoxygenated blood and pulling that into the alveoli, where the partial pressure difference is 46 to 40, much less than that of 140 for oxygen. And again, employing Henry's law, where we can easily pull more CO2 out of that with a lower partial pressure gradient. So, in effect, what I'm trying to describe is that when we move from the arterial side to the venous side, we're going to pull oxygen from the alveoli to bring it up to 100, and that is going to represent a PO2 of 100 is going to represent our oxygenated blood. This is oxygenated. This is deoxygenated. And our partial pressures are ranging at rest from 40 to 100. Does that make sense? Okay. I would stress that you guys get acquainted with this, because we're going to be looking at, and I realize now I will save the oxygen saturation. You'll be looking at the oxygen saturation curve. We're looking at a PO2 of 100 and a PO2 of 40. And we're going to start to look at the effects of not just oxygen, but looking at the effects of CO2, heat, and the effect of hydrogen ions, and how that's going to affect our hydrogen saturation curve. So in order for this to make sense on Thursday, you got to have a good concept of all this right here. Okay, the partial pressure ranges from 100 down to 40. And how that's going to affect our hemoglobin unloading oxygen. So with that disclaimer, what questions do we have? Feel free to ask me when we start on Thursday. All right, so now that we have that in place, let's take a look at the ventilation perfusion coupling. Again, that concept that I mentioned earlier, the second point concept. We've explained the partial pressure difference, which is going to affect the rate of gas exchange. Let's take a look at ventilation perfusion coupling. And this is all related to autoregulation. This is the first slide that I started off with that talks about what happens in hypoxic, low oxygen, or hypercapnic, high CO2. And in alveoli, where we get reduced alveolar ventilation, again, somebody who's doing very high respiratory rate, low tidal volume breathing, so So somebody that's doing that is going to get hypoxic, meaning bringing the PO2 in the alveoli to, we're lowering the partial pressure of oxygen in the alveoli. At the same time, raising the partial pressure of CO2. What happens with the perfusion that takes place is that the pulmonary arterioles are going to cause constriction. So we see constriction in the precapillary sphincters that's going to reduce the amount of blood flow. This is very similar to in concept when we talked about what happens with the sympathetic response. I want to increase blood flow to our active tissues, and I want to decrease the blood flow in areas that need less blood flow. So in effect, what I'm doing here is reducing the blood flow to areas that are hypoxic or hypercapnic. And that should make sense. I want to reduce the amount of blood flow because this is going to be wasted perfusion. This is blood flow to an area that's not going to be participating in gas exchange. So I want to redirect that blood flow to the other healthier areas. So now I'm going to take what's going to happen in well-ventilated alveoli. So areas that are going to have high oxygen content and low CO2 content. So somebody who's taking long high tidal volume breathing at a lower respiratory rate. So what I want to do in this case is if I've got a very well ventilated alveoli, I want to increase the blood flow. And the way we can do that is to dilate the arteriole and open up the precapillary sphincters to increase perfusion into that capillary bed. Increase the amount of perfusion to pick up that rich O2 and also unload a good amount of CO2 into that alveoli. So better ventilated areas of the lung will want to increase the blood flow. Poorly ventilated areas of the lung want to decrease the blood flow. That's the easiest way to explain the slide. Want me to say it again? Yeah. So well-ventilated areas of the lung want to increase perfusion, while poorly ventilated areas want to decrease perfusion. We want to make sure that those are coupled nicely in order to optimize the amount of gas exchange that takes place. The movement of air as well as the delivery of blood flow to that area. Now this slide is going to pinpoint, and I'm just going to cover O2 transport, CO2 transport, bicarbonate buffer, and yeah, and I'll stop there. So I got a couple more slides to talk about before I let you guys go. This entire pathway may look a little scary, but in essence, it really isn't all that scary because if you look at it, the numbers just kind of recycle back and forth. So I would urge you guys to just look at it when you're studying and just follow the patterns, but I'll dissect it for you right now. The PO2 and PCO2 partial pressures of O2 and CO2 during respiration, we're going to account for external respiration up at the top, internal respiration below. And this is going to account for the circulatory system, which is going to be pushing blood through. Now, this is going to talk about the movement of gases down their partial pressure gradient. So we already know that the movement of a gas is down its partial pressure gradient. So we move from atmospheric air over to alveolar air, just like I've drawn on this poorly designed drawing that I did earlier. This is atmospheric air. By the time it makes its way into the alveoli, it's going to mix with the stale air. So we're going to see a decrease in the O2 and an increase in the CO2. By the time we make it from atmospheric air to the alveolar air is going to have a partial pressure of 100 and a PCO2 of 40. The pulmonary arteries that are supplying deoxygenated blood are going to have a PO2 of 40 and a PCO2 of 46. So given that, by the time we make our way over here, we know that the direction of O2 is going to move into the capillaries, while CO2 is going to move out from 100 down to 40. So right here should be a PO2 of 40 and a PCO2 of 46. And I'm just going to move down the respective gradient, participating in external respiration, or rather not participating, but representing external respiration. Pulmonary veins are now going to carry, say we move from a PO2 of 40, which is deoxygenated blood, and we're going to equilibrate up to 100. Just like we mentioned on this slide, we're going to go from 40 all the way up to 100. By the time we make it to about a third of the way, we're going to be fully oxygenated and we'll be matching that of alveolar PO2. So a PO2 of 100 and a PCO2 of 40. And that's going to be pushing all the way down the arteries, through the systemic circuit, through the arteries, and those systemic arteries are carrying oxygenated blood at 140. By the time we make it over to our cells are going to be hypoxic, meaning they're using that O2 and they're pushing out that CO2. So naturally, the PO2 is going to be 40 and the PCO2 is 46. So the direction of oxygen is to move into the cell, and CO2 is going to move down its gradient from 46 to 40. That's internal respiration. Once we get over there, it's going to be the reverse. PO2 of 100, by the time we make it about a third of the way, PO2 is going to be about 40, and the PCO2 is 46. Again, because of our high PCO2 in the tissue, we're going to get that unloading of CO2 into the capillaries, and the capillary is going to pick up that excess CO2 to make its way back over to the lungs, where it's going to, again, push out CO2 and pull O2 from the alveoli. So that whole circuit just goes back and around. It just illustrates the entire pathway that we learned. Everything from the systemic circuit, from the left atrium to the left ventricle out through the arteries, through the capillaries to the veins, back to the right atrium to right ventricle, through the pulmonary arteries, back over the pulmonary capillaries to participate in external respiration. And we just go hand in hand to exchange O2, CO2 up in the lungs and exchange O2, CO2 on the tissues.
So the systemic has higher pressure because it goes to the body, that's why the values are bigger?
Yeah, at this point?
Yeah.
Yes.
And then
It matches that of alveolar.
And then the deoxygenated, it's lower, right? Because it's going back to the pulmonary.
Yeah, this is returning back because it was able to exchange with the tissues.
That's
And
why
now
it's
you've
lower,
got,
right?
Yep, this is basically your leftover O2 and your excess CO2.
The values never change?
Hmm?
The values never change?
They do actually. This is at rest. So what I'm illustrating right now is just a simple idea of what's happening. So think about it this way. Your oxygenated blood is carrying a good amount of oxygen. You're supplying that oxygen to the tissues. Whatever is left over is going to make its way back, returning into through the pulmonary circuit and picking up the remaining oxygen. It's going to top off back over here, and it's going to make its way over. So the entire circulatory system is just picking up, topping off O2 up in the lungs, and it's going to unload that O2 back where it's needed. So what happens if you have tissues that are using more oxygen? We expect the partial pressure of O2 to be drastically lower. So let's say we bring a PO2 of, and we'll talk about this on Thursday. PO2 we'll say is at 20, and the PCO2 will be about 50. That's going to increase the partial pressure difference from the systemic arteries at 100 to that of 20. So now I've increased that gradient. That means I'm going to have more oxygen unloaded into the tissue, and my partial pressure of O2 leaving is going to be about 20, and then I'm going to increase the partial pressure gradient taking place in external respiration. So this is our way of picking up more O2 and unloading excess CO2 when needed by increasing the partial pressure gradients. What questions do we have about that? So think about that concept of our circulatory system unloading O2 and topping off in the lungs. That's exactly what we're going to be talking about when we look at hemoglobin unloading. So with that, what I'm going to leave you guys with is how we are able to transport. We've been talking about the partial pressure of O2 and CO2. Now, we're going to now talk about in the next slides how we carry that O2 and CO2 and how that's going to tie in on a cellular level, even down to the molecular level with our molecule hemoglobin. As I mentioned, hemoglobin, as one of our quiz questions, hemoglobin is the protein that carries oxygen within the red blood cells. And here's our big red blood cell. We've got 250 million hemoglobin molecules in this red blood cell. So the purpose of our hemoglobin is to carry oxygen as well as carry CO2. And it just depends on what we have more of. And that's the vague concept I'm going to leave you with that will segue into what we talk about on Thursday. In oxygen-rich environments, hemoglobin will pick up oxygen. In carbon dioxide-rich areas, it's more likely going to bind to carbon dioxide than it is oxygen. So, in effect, hemoglobin, or oxygen and carbon dioxide compete for hemoglobin, whoever we have more of, whether it's more oxygen or more carbon dioxide. Right now, I'm just talking about oxygen, where in oxygen, out of all the 100% of oxygen that we can carry in our circulatory system, 1.5% of that is dissolved in gas form. And that's just dissolved right there in the plasma. The other 98.5%, since we have low solubility of oxygen in the blood itself, 98.5% has to actually be carried by hemoglobin. When hemoglobin binds to oxygen, we call that oxyhemoglobin, or represented by HbO2. So the way that works, in the alveola, we have high O2, and it moves down with partial pressure gradient of 100 down into the interstitial fluid. Through the interstitial fluid, it's going to diffuse across the capillary wall through the endothelium into the plasma, of which 1.5% will be dissolved in the plasma. The remaining is going to get picked up and diffused into the red blood cell, where hemoglobin, which is initially bound to a hydrogen atom, is going to kick off that hydrogen atom and bind to the oxygen molecule. So naturally you're going to ask, well, what do we do about that hydrogen ion? What's going to happen with that? That's going to play another role in just a little bit. Okay? So we kick off a hydrogen ion. Hemoglobin is now called oxyhemoglobin because it's picked up that O2. So as it picks up O2, it's going to continually pick it up from the plasma, and then whatever's left, about 1.5% of that is freely dissolved. The next thing is to look at CO2 transport. How do we carry carbon dioxide? And there's 3 ways in which we can carry it. There's 2 ways for oxygen. There's 3 ways for CO2. We know that CO2 dissolves better in a liquid than it does oxygen. So 7% is dissolved of that CO2 in your entire system is dissolved in the plasma, right there in the blood. The other 70%, actually let me talk about the 23%. 23% is going to be attached to hemoglobin. So some of your hemoglobin is going to be carrying carbon dioxide. And as I mentioned, hemoglobin will compete with either O2 or CO2. If there's more O2, we'll get oxyhemoglobin. If there's more CO2, we're going to have deoxyhemoglobin. So looking at that, 23% being carried by hemoglobin, the other 70% is going to be converted into carbonic acid. That means whenever you get CO2 and water, anytime you get CO2 in an aqueous environment, CO2 and water combine into a weak acid. And that weak acid is called carbonic acid. This equation right here, I know we touched on in our cellular physiology lecture, I believe. Or no, our biochem lecture. In our biochem lecture, we talked about that. This is now, we're going to bring this equation into place, because you're going to be seeing it quite a bit in our respiratory lecture, and you can even see it in our digestive lecture, as well as our urinary lecture. So CO2 and water mixed together, you create a weak acid called carbonic acid. And carbonic acid can disassociate from one another, creating a hydrogen ion and a bicarbonate ion. That bicarbonate acts like a buffer because it can also pick up hydrogen ions. So what do we do with this other hydrogen ion? Well, this is going to get kicked off and it's going to get excreted by the urinary system. We have a way to regulate the amount of hydrogen ions, but we get rid of hydrogen ions in order to create bicarbonate. This bicarbonate is the buffer that prevents the pH changes in your blood to keep it between 7.35 to 7.45. So with that bicarbonate that's going to be used, we're carrying that CO2, that excess CO2 that we're carrying, we use that as a buffer. So it's a way to nicely conserve that CO2 and give it a purpose other than just some metabolic byproduct that we're trying to get rid of. It's one of the real intricate designs of the human body. So with that, that's where our CO2 comes into play. We'll talk about how that plays a role in unloading of O2 with the competing CO2. The last slide I want to leave you guys with is a bicarbonate buffer. So how do we create this bicarbonate buffer? Our CO2 and H2O, again, when we increase our respiratory rate or decrease our respiratory rate, is how we account for the amount of CO2 in our blood. The CO2 that gets dissolved in water will create that carbonic acid. That carbonic acid will again dissociate into a hydrogen ion, where we can either remove it or we can add it if we need to. We've got other buffer systems like the kidneys that will be able to eliminate those hydrogen ions. That buffer, that HCO3, it becomes part of our bicarbonate reserve. So in the cases where during cellular respiration, you don't have enough oxygen to supply to your muscle tissue, because we don't supply enough oxygen, we're going to start creating that pyruvic acid into lactic acid. That lactic acid is going to have a hydrogen ion that's attached to it that can eventually make its way into the capillary and effectively reduce the pH of the blood with the introduction of those hydrogen ions. We want to keep that blood between 7.35 to 7.45. Otherwise, we start to denature some enzymes. We start to cause a whole host of things. So that bicarbonate that comes back out will come in and buffer those hydrogen ions. Because if we get hydrogen ions that come in, those two bind together, HCO3 and hydrogen ion, those buffer together to create carbonic acid. Carbonic acid can dissociate back to CO2 and H2O, and then we can increase our respiratory rate to get rid of CO2. So in essence, that's what you're doing when you are doing some form of exercise, or let's say you're running to your car after you're done with lecture. When you're in your car and you notice that you're breathing a little bit heavier, you're increasing your respiratory rate and also effectively increasing your tidal volume to blow off that excess CO2. Because that CO2 that came in is not only picked up, going to be sent over for external respiration to exchange that oxygen, exchange that CO2 for oxygen, but that excess CO2 is also helping buffer the hydrogen ions that are present in your blood as a response to anaerobic metabolism. So again, part of the intricate design behind it. With that in mind, now that you have that, we'll play on that a little bit more. We'll talk about the production of hydrogen ions. So explore those concepts. Make sure you get brushed up on this whole concept, because we're going to take this into oxygen loading. How do we get hemoglobin to unload oxygen? We're going to take a look at the oxygen and hemoglobin saturation curve and look at it in hypoxic conditions and see how we can get hemoglobin to unload O2 and pick up O2 in both the lung as well as the tissue. So thank you again for your guys' attention. I will see you guys on Thursday.